Yes, welcome to the third, I think, GA4GH 101 webinar. I'm Angela Page. I'm Director of Strategy and Engagement at GA4GH. I'm based at the Broad Institute in the US. And I'm here with Jeremy and Nirja, and the three of us will be delivering today's presentation. Okay, so we're going to use something called Slido for participation in Q&A, and we really would like this to be as interactive as possible. So throughout the talk, throughout the conversation, you can go to sli.do in your web browser and type in GA4GH 101 or scan this QR code and type in your questions as they come to you, and we'll have a couple of points throughout the session to stop and answer questions. You can also, if burning question right in the moment, you can raise your hand or ask it in the chat and someone will get to it. We, like I said, have a number of folks from staff here today, so there will be plenty of us to answer questions as we go through. But to start, we're going to use Slido for a little icebreaker activity and just to kind of get to know each other and also to get to know the platform. So if you go over to sli.do, again, type in GA4GH 101, you'll have a question, where are you joining from? And you can just type in wherever in the world you're coming from, which would be lovely to see where we're all joining from. And thank you, Nirja posted the instructions in the chat. We've got quite a few from Canada, and yeah. Australia. So I am really glad that you're here. This is our first time doing it at this time zone, and the whole idea was to try to get more folks from a wider cross section of the globe. But we've still got our Brazil, Germany, great, cool, thank you. San Francisco. So it's always fun to do this just to see how global of a community we have joining these calls and having interest in GA4GH and participation. We're always trying hard to get the first G in GA4GH better, bigger, more global and global alliance for genomics and health. All right, so I'm going to move on to the next poll, I think, if I can. So just tell us why you're here, what you're interested in learning about. And if we can, we will talk about it. And if not, we will follow up. And if you really, really, really need to know something and you want to make sure that we get it, you might put your name on it so we could follow up with your name just in case we don't get it today. Or you can always email us at secretariat@ga4gh.org. So you can pop in what you're interested in learning about. Are you interested in the structure of GA4GH, our mission, why we're here, what we do, how you can get involved? There you go. Are you interested in a particular work stream? Have you heard about any of our standards and work streams already and want to learn more about something in particular? Or you've never heard of GA4GH before and you just kind of stumbled on this Zoom link? Could be. All right. I've been told to wait 10 seconds before moving on to the next thing, but I can't. So here we go, we're moving on. How are you currently engaging with GA4GH at this point in time? So have you already joined a mailing list? Are you participating in one of the work streams? Are you brand new? Have you attended a plenary meeting but haven't gotten involved since then? Again, I'm not sure how you'd be here if you had never heard about GA4GH before, but so there's one of you at least out there. Okay, we'll have to find out how you found us. All right, so a number of folks subscribe to the mailing list and probably maybe get our weekly newsletter, and we kind of flood your inbox every now and then, and you're trying to figure out what all is going on here, and we can hopefully set some things straight for you today. Excuse me. All right, I think that has steadied out, so thank you. Keep your questions coming. You can add them to Slido as we talk, as I said, and we will stop a couple of times during the conversation to answer questions. All right. And just if there are questions, they'd show up here. There are none yet. That's fine. All right, so let's dive in. The Global Alliance for Genomics and Health is the standards organization for genomics, the International Standards Organization for Human Genomic Data Sharing. We were founded in 2013 with a mission to accelerate progress in genomic science and human health by developing standards and framing policy for responsible genomic and health-related data sharing. So we have four kind of interrelated aims, which are to enable international data sharing, but also to encourage technology-enabled federated approaches to sharing where, it's not possible to aggregate and use sort of traditional data sharing approaches. We are taking this technology forward-looking approach, which Jeremy will tell you more about later on. And we promote sharing and interoperability across the translational continuum, meaning that from the moment of the point of care or the research participant, we talk about, we have things, standards and policies for that aspect where it's, collecting consent and interacting with the participant through to healthcare research back into this global learning health system, which Jeremy will also talk more about later. But the idea is that GA4GH really is promoting sharing across that whole landscape. So this is the GA4GH ecosystem. We have over 650 organizational members now across 90 countries. So an organizational member is just an organization of any kind. It could be a commercial company, a research institution, a library, I don't know, anything that has committed to the mission and principles of GA4GH. So they've signed on to this global data sharing mandate. We also have 4,000 plus, probably much more than that by now, subscribers. So these are individuals who have, like some of you on here, subscribed to our newsletter, but haven't necessarily, they're just sort of observing, watching, seeing what GA4GH produces. Of those, about 450 or so are active participants in the GA4GH work stream. So these are the actual, the folks that are boots on the ground, having conversations every week about how are we going to standardize this activity and these sharing efforts. They do that in the context of 8 work streams, and we have 24 what we call driver projects who sit on those work streams, and I'll tell you more about that as well. But together that community has so far produced 20 technical standards and 9 regulatory policies and frameworks. And we're aware of 40 plus implementations and 40 plus institutions that have implemented or deployed our work in their local context. And several publications. You also note that we had a special issue in the new journal Cell Genomics, where you can learn a lot about GA4GH and what we're up to. If you want, we can share that link in the chat probably. All right, so back to driver projects and work streams. So I think this is a common question that folks have about GA4GH. What is the difference? So, and we also like to talk about our matrix organizational structure, which we're very proud of. So this is the GA4GH matrix. Like I said, we have these 8 work streams, and those are the horizontal rows that you see here. And then we have 24 verticals, 24 driver projects. So the driver projects are these real world initiatives external to GA4GH, things like Australian Genomics, ICGCRGO, Genomics England, ClinGen, that have existed long before GA4GH, but have a particular interest in realizing our mission. And they have committed to contributing resources to GA4GH and to both participating in the work streams and piloting the products of the work streams. The work streams are internal to GA4GH. They are the bread and butter of GA4GH. They are the workhorses, the groups that do the work of creating the standards. I'll go back a slide and show there's 2 foundational work streams, regulatory and ethics and data security, and they really underpin everything else that GA4GH does across the technical work streams that are listed here. I won't go into them, Jeremy will do that later. But they also produce their own work. And then we have this column on the right side, partner engagement, which is about making sure that what we're doing is, we have bi-directional communication with the broader community. So I'm just going to keep going here. So this is our driver project community. Like I said, we have 24 driver projects with representation around the globe. Some of that is direct, some of it's indirect, meaning the headquarters might be in South Africa, in the case of H3 Africa, but that is a continental network. And so we, when the network promotes a GA4GH standard, the idea is that it sort of propagates across that community. That's really the sort of strategy we have for engagement there. And then similarly, there's a group called the Genomics and Health Implementation Forum, or GIF. This is the group that implements GA4GH standards in the real world at the end of the day. So similar to driver projects, where they have committed to piloting standards, the implementation forum is an opportunity for initiatives to come together and share resources, share experiences in implementing GA4GH standards, and then feed that back into the work streams to improve it. This is that blue column on the right that I was talking about before. And excuse me. These are the current GIF members. So the GIF started out as a group of national initiatives led by Australian Genomics and Genomics England, saying, these are national genomic data initiatives, setting up a clinical genomics infrastructure in many cases, and saying, how can we learn from each other, and how can we come up with common practices so that we can share data across our initiatives. But over time, I think we have also started to open up beyond just national initiatives as well. So with all of that in mind, the 24 driver projects, the Genomics and Health Implementation Forum, we know that we're still not hitting every single global genomic initiative out there, or every single genomic data initiative around the world. We're aware of at least 200, and I'm sure there's more by now, initiatives. And so the idea with the driver projects isn't that we have representation from every single group out there, but that we at least have representation, so that we're hitting all of the sort of the buckets and making sure that we have representation. Together, these many initiatives are generating huge amounts of data. A few years ago, we had a review, or an article in it was a preprint in bioRxiv, where our chair and some others estimated that by 2025, there would be 60 million human whole genomes generated in healthcare available for research. We have yet to determine whether or not we made good on that estimate. We have a couple more years to go. But the point is that where historically genomic data was being generated in the research sector, we're starting to see a shift into healthcare and clinical trials and cohort initiatives. At the same time, we're also starting to see an increase in the proportion of genomes that are being stored in the cloud. And as you can see here, this is a projection, but we're expecting that to just sort of skyrocket over the next few years. So the opportunity embedded in all of this is that if we can make secondary use of that clinical genomic data, and we can connect it to research data, and we can use GA4GH standards to bring it all together, we'll have a virtual cohort of at least 60 million samples by 2025, from which researchers can learn and then contribute to improved healthcare and patient outcomes. And so from here, I'm going to give it to Jeremy, who's going to get much deeper into the work streams and this global health continuum thing. So take it away, Jeremy.
Thank you, Angela. Hi, everyone. I'll just give a quick intro. I'm Jeremy Adams. I'm a senior software developer with the Global Alliance for Genomics and Health. I'm based out of the Ontario Institute for Cancer Research in Toronto, Ontario, Canada. And so I lead the core staff development team. We work on things like improving documentation and to the standards and standards improvement, as well as reference implementations and testing applications for the standards themselves. Okay, so let's see if I have control. There we go. So yeah, the take-home message essentially with our ability to sequence many more genomes and have better approaches to understanding and analyzing and sharing those genomes, is that, if we create a cohort of properly consented genomic data across the globe, we'll enable a host of downstream impacts for research and precision medicine, including more powerful analyses and stronger variant interpretations. At GA4GH, we've identified 4 major approaches to data sharing, which are through centralized genomic knowledge bases, through trusted repositories of multiple data sets, known as data commonses, through hub and spoke models of federation, where there are standard APIs to facilitate communication between all the different systems within a federated network, and also a linkage of distributed data, again, through standards-based technology solutions. And GA4GH recognizes that all of these types of resources and all these infrastructures will be important for the next generation of genomic analysis and data sharing. So we're seeing a shift from an analysis model of downloading large data sets to local machines, environments, and compute clusters. We're transitioning towards an environment where analytical workflows visit the data where it lives originally via standard APIs, and analysis is conducted on the data in its original place. So we see this model of federated analysis in which multiple different machines and computers are talking to each other within a network, and analysis is happening across multiple nodes. We see this type of model as dominating the genomic data sharing landscape going forward. Federation enables a number of advantages. It allows the data to remain in its location of origin and provides and for us to provide broad access methods to the data, which respect the regulatory processes of that region and organization. And if we can enable a federated system between healthcare and research, it will allow us to transfer methods and skills into healthcare and leverage the healthcare data for secondary research. So this model of continuously adapting a system by feeding data and learnings between healthcare and research is supporting what we call a learning health system. And this is one way to think about our work in action and how it fits into that context. A learning health system is our term to describe a continuously adapting environment in which data and analytics generate knowledge. That new knowledge is implemented into practice for the betterment of patient care. These new practices change the landscape and generate new and better data. And then this feeds back into the system to lead to new research and to generate ever new knowledge, creating an ongoing virtuous cycle. So in this cycle here, we're showing one manifestation of a learning health system as hypothesized data lifecycle, beginning with obtaining consent from the data donor or patient, moving through to responsible data storage and access, supporting secondary research in a federated network, and all leading towards improved knowledge of informing health and medicine. So you'll see here that each of the standards produced by GA4GH can fit into one of these steps in the cycle. It's important to note that this is just one example of how GA4GH's standards and resources can work together. They are extremely flexible and can be suited to a wide variety of research contexts and data life cycles. And so I think with that, we'll take a quick pause on the presentation and maybe move over to some audience questions if there are any.
So I answered this first one, Jeremy, in the Q&A, but I don't know if you have anything more specific to say. So the question is, do any of the work streams address challenges or solutions around anonymization or de-identification? And this relates to our data security work stream. I'm not aware of anything specific, but Jeremy, do you know of any specific standards activities there?
I would just add that, yeah, it's mainly it's really the foundational work streams that deal with how the data is to be collected and shared across other research environments in a properly anonymized, de-identified way that's also very secure and that retains the security of the data being shared. So obviously we're talking about a network where data is sort of flowing through a network, but that doesn't mean that it's open for everyone. There are still a lot of authorization and authentication mechanisms involved. And so with all of our standards, there's a sort of review process on both the security and regulatory and ethics side to highlight any potential issues that there might be with re-identifying patient data and other types of those human rights concerns. And so we always make sure that when we develop standards before they get officially approved, that they've gone through this rigorous check of not leading to those kinds of problems.
Mm-hmm. Yep, and I think there's also a number of folks on the workstream who are interested in that and sort of the more technical research aspect of that. And if something were to come up, I think we would be interested in bringing it into the workstream. How does GA4GH engage industry? If the big players aren't using these standards, it might be challenging for them to become widely adopted. We completely agree. We are very lucky to have quite a bit of participation from Google and increasingly from Amazon Web Services and Microsoft and the Broad Terra system, which is kind of connected to all three of those, is also participating in GA4GH. Jeremy, do you have something specific to say here? I think it's a valid concern.
Yeah, absolutely. I would just build on that, Angela. The cloud work stream, as we'll get into the depths of it later in the presentation, but they are essentially developing workflow standards, so systems and standards for executing analytical workflows for genomics in the cloud. And so as part of that, the major cloud providers, AWS, Microsoft Azure, and Google Cloud, are all implementing one or more of those GA4GH standards within their own environments. So there is sort of a plan to have maybe like a native workflow execution service, which is a GA4GH standard in some of these cloud environments. So that's one aspect of the engagement of industry. Another side of it might be in the clinical work stream, where we are looking to get our standards like phenopackets more involved in standards that are more widely used in healthcare, and then also thinking about how that extends to EHR systems. So yeah, it is a big goal of ours to get our standards into industry, and we are always looking for more and more partnerships to bridge those gaps. Yeah.
And I would also mention Illumina has also been heavily involved since the beginning. I think your point about clinical and phenotypic data capture and what industry engagement looks like there is different from what industry engagement looks like for the cloud work stream. And so there isn't one blanket solution. I think we have tried to have sort of industry type groups, but it's such it doesn't mean one thing. And so that'll be a very context specific solution. Are you planning on adding more work streams? At the moment, no, I don't believe so. But I think there's definitely opportunity and room for growth within GA4GH, even within the context of the work streams that we have right now. And I think that if something really major came down the road, we would find a place for it. Do you agree, Chairman?
Yes, yeah.
All right, so maybe we pause or stop questions and we'll come back to them again. There will be another time for more questions later on. So Jeremy's now going to really dive in.
All righty, so yeah, let's get into it. So here are our 8 work streams. As it's been mentioned earlier, there's 6 technical work streams and 2 foundational work streams. The foundational groups are data security and regulatory and ethics. We call them foundational because they sort of provide this working model and this foundation upon which all of our other technical standards and work streams are built. So I think we begin with ClinPheno. So the clinical and phenotypic data capture work stream, or ClinPheno for short, supports clinical adoption of genomics by developing informational models and standards for describing clinical phenotypes in genomic medicine and research, including the capture of information between clinical research and patient-centered systems. So there are 2 main subgroups or focus areas. One is phenopackets, which is a standard for phenotypic information. So while there are standard formats for the exchange of sequence and variation data, such as the BCF format, until recently, there hasn't been any de facto standard format for the exchange of phenotypic information. Some initiatives and databases have defined their own formats, but because those formats have been developed for particular systems, there's a lack of uniformity that hinders downstream communication and interoperability. and it limits the ability to perform analysis across multiple systems. So phenopackets is meant to fill that gap. It's an exchange format for phenotypic information that improves upon, maybe phenotypic information only being relayed in free text. And it's supposed to be, it's very flexible as a JSON structure, and it's not quite as complicated or heavyweight as like an entire EHR. The latest version is phenopackets version 2.0. It was approved in June, and it's a stable version that is now being implemented by a number of implementers worldwide. The pedigree standard is intended to be a computational exchange format of family health history, as well as a representation for larger, more complex families and relationships. It's a graph-based model intended to align with other structures and standards like HL7 FHIR, as well as it integrates with phenopackets. You might be thinking already that there are already pedigree standards like the PED format, but PED, within the working group, there are some shortcomings identified with that format in that it can't represent complex relationships more than a trio. It doesn't capture things like adoption, donors, pregnancy, vital status, multiple phenotypes, and all of that type of data is very important for genetic counseling and risk assessment. You need these sort of richer relationships to be able to describe those things effectively. So the team has a draft model developed, and they're currently working on testing, refining, and implementation. It's for this standard, it's a great time to get involved and review the work that's been done to date. So if you're interested, well, at the end of this presentation, there'll be contact information for all of the various work streams, and you can see which work stream is most of interest to you. Not shown on this slide yet, because it's not quite on the roadmap officially, is the computable cohort representation group. And this group is exploring the space of cohort discovery, extraction, and comparative statistics. And they're in the early stages of defining potential standards. So next we'll move on to the regulatory and ethics work stream. As I mentioned, it's one of our foundational streams. So regulatory and ethics, or RUES for short, focuses on ethical, legal, and social implications of international data sharing. The work builds upon a human rights framework to create and harmonize forward-looking consent and privacy policies and anticipatory data governance models. So just to highlight a few of the major deliverables from RUES, there's a policy on clinically actionable genomic research results, which is a policy that aims to provide a reference point for managing the return of results that recognizes the importance of accountability and transparency of genomic researchers toward participants. There's the engagement framework, which provides considerations and questions researchers should be asking when engaging participants, patients, and the public in genomic research and health implementation. There's the DACRES, which has recently been published, stands for the Data Access Committee Review Standards. And these are procedural standards for data access committees to facilitate consistency, effectiveness, and robustness of reviews for data access requests to genomic and health data. RUES is currently finishing up their last few deliverables on their roadmap, and they will be calling on the community to provide new topics to explore that are of vital importance to genomics within a human rights framework. We've been discussing things like artificial intelligence and machine learning bias and its implications in genomic research. There's also some next steps planned for the Data Access Committee standards. And again, if there are any topics that you'd like to explore or interested in learning more about, please get in touch, and that information will be there at the end. Okay, so now we'll move on to the Data Use and Researcher Identities work stream, or DURI for short. So the overall aim of the DURI work stream is to enable researchers to efficiently discover genomic data and then apply to get access automatically based on their digital identity, and if required, based on machine readable research purpose ontology and vocabulary to access certain data sets with restrictions about their use. So this overall aim is enabled by three main DURI standards. First is the GA4GH passports. Passports is a standards-based digital identity for researchers that facilitates their access to distributed data across various systems and platforms. The passport includes ways to express rights to data access for fine-grained permissions and specific data sets. The passport contains what we call visas as evidence of access permissions that have been granted by a data owner or institution. And that permission can be carried across institutions for different data environments. So facilitating access to data that might be located elsewhere than the original authority provider. Secondly, there's the data use ontology, or DUO for short. And DUO allows data custodians and providers to semantically tag their own data sets with restrictions about their usage, such as, this data can only be used for cancer research or for nonprofit research. And this vocabulary makes it possible to automatically discover data sets based on the authorization level of users and their intended usage and what they plan to do with that data. Lastly, the machine readable consent guidance was developed in collaboration with the regulatory and ethics work stream. It describes how to adopt data sharing consent language that unambiguously maps to the DUO terms. This facilitates data access request submissions and approvals in a much more streamlined and efficient manner. Next, we have the data security work stream. So again, data security is the other of the two foundational work streams of GA4GH. And data security creates technology standards and best practices for protecting data and services that are consistent within the GA4GH human rights policy framework. This includes the development, customization, and adoption of standards for identity management, data security, patient and data donor privacy protection, and assurance of the service. So specifically, the data security deliverables include one example is the risk assessment methodology for software stacks. This standard aims to provide a guide in assessing risk for those in the genomics community and how to define what information and data needs to be protected. There's the cloud security and privacy policy, which aims to develop specific guidelines and recommendations for secure and privacy conscious use of cloud services and the considerations when patient data might be present in the cloud. Then there's the AAI standard, or the Authorization and Authentication Infrastructure Standard. This is a technical profile for managing and authenticating the identity of users and for authorizing data access requests. AAI also links back to the DURI workstream by incorporating research identity vocabulary developed by DURI and provides protocols on how to transport that information contained in passport visas. So the key takeaway of AAI is one of its major use cases is actually in the passport standard. So the passports standard is built upon this authentication and authorization infrastructure framework. Next, we have the discovery work stream. And so discovery aims to address challenges of discovering and utilizing genomic data that's increasingly abundant, but it's also disparate in the way that it can be represented across a wide array of systems and institutions. So the discovery workstream develops standardized APIs and context-aware user interfaces, aiming to create a unified data discovery framework that makes it easier to find and use data, as well as tools and infrastructure for subsequent genomic and clinical analysis. The major standards of discovery include the Beacon API. Beacon is a federated web accessible service that can be queried for information about a specific genomic variant. And that query returns a simple report on its existence within the queried database or resource or website. Beacon version 2 is currently in development, and it will include additional support to securely share clinical information alongside a variant, such as diagnosis or significance associated with a variant. Another standard, the Data Connect API, it's a standard that addresses the challenge of publishing and discovering biomedical data. This data is often stored in a variety of locations. There are many different methods, formats, and file types associated with accessing this type of data. So Data Connect consists and makes use of a common JSON schema paradigm in which data providers can describe their data using Data Connect methods without complete retransformation and reharmonization of their underlying data into a new framework. So Data Connect also consists of methods for searching for that data across multiple services using custom queries. Lastly, there's the service registry and service info APIs, and these are kind of fundamental to all technical APIs across all GA4GH work streams, not just within discovery. But service info and service registry provide a networked infrastructure for all API-based GA4GH standards and services to create an internet of genomics in which it's possible to discover new services based on the information that's present within registries of those services, and also to learn more about the type of data that might be served by any particular service using the data that's relayed through the service info endpoint. Next, we have genomic knowledge standards. So many genomic analyses require identifying variation in genomic sequences that are potentially causative of changes in phenotypes, such as the progression of disease. The Genomic Knowledge Standards, or GKS workstream, develops standards-based components to enable unambiguous exchange of reference genomic information through common APIs, thereby enabling downstream analysis and interpretation of genomic data. Standards developed by GKS include the Variant Representation Specification, or VRS, which is a standard representation of many different forms of genetic variation, like copy number variation, haplotypes, allelic variation, such as single nucleotide variants, or SNVs, and indels. Another standard known as the Variant Annotation Specification, or VA, is a standardized schema that focuses on interpreting the observed variants with details about how they may functionally impact gene function and phenotype. For example, how they may lead to the progression of a disease, or how effective a particular therapeutic may be given a molecular profile of variation. Next, we have the large-scale genomics workstream. High throughput next generation sequencing technologies continue to produce data at a massive scale. That scale is still accelerating. So as sequencing technology improves, we can get it to greater and greater depth, and that just makes the data footprint larger and larger. So the large scale genomics workstream is, their aim is to meet this challenge through efficient and standardized formats to store, access, and analyze sequencing reads, genetic variation, and gene expression information. The LSG workstream envisions the landscape in which traditional file-based approaches to accessing that information is no longer scalable, and instead researchers must use standardized API interfaces for being able to assess the data effectively. So you can think of the large-scale genomics standards falling into two broad categories, one being file formats and the other being APIs, and they're related to each other, but they're distinct. So to get into the file formats, there's the sequence alignment map, or SAM format, which is the industry standard tabular data format for aligning raw read information from a sequencer to a reference genome. We also maintain and develop the binary counterpart of SAM, which is known as BAM, as well as CRAM, which builds upon BAM again to as a compression standard. So this is for long-term compression of genomic alignments. The variant call format, or VCF, is the format for cataloging and annotating instances of genetic variation, such as SNVs and indels. There's also a standard called GA4GH, which is an encryption layer for private and sensitive genomic alignments, both in transit and at rest. It can be used in conjunction with CRAM for both genomic compression and encryption of the data. Moving into the API side of the large-scale genomics workstream, there's the HTS-GET API, which is for streaming high-throughput sequencing alignments. So essentially alignments and variants, or CRAM files and VCF files. It's an API for streaming that data, but the researcher is allowed to specify targeted regions that are of interest to them. So not relaying the entire file necessarily, but relaying regions of genomic regions of interest. There's the RefGet API for streaming reference sequence data. And there's also the RNA-GET API for streaming RNA-seq gene expression matrices. Similar to HTS-GET API, RNA-GET can return dynamic matrices that only contain the genes and the tissues or treatment conditions that the researcher is interested in studying further. Last but not least, I believe, is the cloud workstream. And so cloud, so modern genomics involves running computational workflows over long periods of time to transform and derive insights from large data sets. An example of a computational workflow is an alignment workflow, or a variant calling workflow, or a gene expression analysis workflow. And so Cloud Workstream develops specifications to streamline how these workflows are launched and monitored over the web, and also how the resulting data is accessed within a network of cloud services. Because genomic data is many orders of magnitude larger than the software used to analyze it, we've adopted a philosophy of bring the algorithm to the data, not the reverse. Therefore, we develop standards to encourage this paradigm of workflow portability and reproducibility. Portability means that an effective workflow can be moved seamlessly to different computational environments, and reproducibility means that a workflow that is executed on the same set of input data should always produce the same set of output data, regardless of the location, regardless of computational platform or the cloud services provider on which the workflow has run. So to meet that challenge, the Cloud Workstream has released a suite of 4 API specifications, and they enable cloud-based genomic analysis that is platform agnostic, so not tied to any particular cloud infrastructure. The Tool Registry Service, or TRS, provides a standardized interface for accessing workflow algorithms. The Data Repository Service, or DRS, allows clients to choose from multiple copies of an existing data set to submit to a workflow based on factors of efficiency. So they can choose the copy of data that is most efficient, considering geographic proximity, or considering the cloud platform hosting the data. There's also the Workflow Execution Service and Task Execution Service, WES and TESS, and they act as interfaces for clients to request that a workflow be run on a data set of interest over the web. And it also provides an interface for allowing the researcher to inspect a particular workflow to see if it's completed, if it's failed, if it's still in progress, et cetera. So moving away from the work streams a bit. So the work streams are where the standards themselves are developed. We also have other groups that are focused on testing out the existing standards to see how they can be put into practice to facilitate real-world federated genomics. So we have the Federated Analysis Systems Project, or FASP. And FASP was created to show that GA4GH APIs can be used together in conjunction with one another to facilitate science and genomic data discovery, access, and analysis in the cloud. So to show end-to-end researcher use cases that are made possible by GA4GH standards. FASP conceptualizes demo use cases that simulate how a researcher would search and analyze genomic data within a GA4GH ecosystem. And so in the past, multiple driver projects and implementers have participated in these scenarios. In 2020, for example, we had a demo in which a simulated researcher requested and executed the same GWAS workflow, that's genome-wide association study. The researcher would execute that workflow on 4 different driver project platforms, and this demonstrated the aforementioned workflow portability and reproducibility. The platforms were all run by separate organizations and deployed on different cloud infrastructure like AWS and Google Cloud. As each platform had implemented GA4GH standards, the client researcher could run the same workflow by using very simple API calls or requests, so that they didn't have to create new methods to execute the workflow. But because the standard sort of harmonized the approaches to running the workflow, it was very simple to run it on multiple instances. FASP also identifies researcher use cases that are not yet feasible given the current state of GA4GH specifications, and these demos perform a critical role in an overall feedback mechanism that highlights what we need to do to improve our standards and make them more usable in the real world. The other side of things is the GA4GH starter kit, which is something that's developed. The development is being led by the team that I run, and these are open source reference implementations. Each application within this suite, known as the starter kit, acts as a server implementation of a single GA4GH API specification. So we've developed it to be deployable in a variety of contexts for local testing and exploration of GA4GH standards, but they can also be scaled up into more sophisticated environments like high performance compute clusters, also cloud-based deployments that we encourage. And so, you can sort of deploy it in a variety of scales and contexts. The starter kit overall will serve an important role in this overall mechanism to improve our standards by providing a reference code base that's easy to update with prototype features. And as new features move from experimental to standardized, the starter kit will keep up with these developments so that there's always reference implementations of the latest versions of our standards so that the community can more easily get involved and understand how to get into the nuts and bolts of GA4GH standards and how to work with them directly. And so I think that's it for me. I will hand it over to Nirja now.
Thanks, Jeremy. Hi, everyone. My name is Nirja, and I'm the workstream coordinator at GA4GH, and I'm based at OICR in Toronto. And I'm going to be answering a question that a lot of you had at the beginning of this webinar, and that's how do I get involved? Well, if you like what you heard about all of our work streams and you have an inkling of which ones you'd like to join, then please contact Secretariat at GA4GH.org and we can give you a little bit more information if you want about some of the work going on in our work streams. And if you'd like your organization to join GA4GH, then you can become an organizational member by visiting ga4gh.org/members. And you can also subscribe to GA4GH updates by going to ga4gh.org/subscribe. Can I go to the next slide or? Oh, no. Okay, great. And I'll also do a quick plug for our new GA4GH onboarding program. As a newcomer, it can be very overwhelming when you join GA4GH, as you might already feel overwhelmed after hearing this presentation. And if you do, that's okay. Our onboarding program connects newcomers like yourself with workstream guides, and our workstream guides are very kind people. They are active workstream members, and they'll help to orient you to this GA4GH community. The program consists of a one-on-one with your workstream guide, and they'll help to direct you toward resources that you can use and let you know how you can participate fully in the standard creation process. If you'd like to participate in that program, you can, again, contact any one of our workstream managers, or you can email me directly at nirja.skantharaja@ga4gh.org. And with that, I'll pass it back over to Angela to start the Q&A.
Great, thank you, Nirja. Such a great program that is new this year and has already gotten a lot of good traction. All right, click to start remote control. Okay, so we are going to continue the Q&A. I think that people have been asking questions over there, which is great. And we're going to let you keep asking questions while we go through the acknowledgements and all of that. So these are our host institutions. As I don't think I mentioned earlier, GA4GH is hosted by 4 international host institutions: Ontario Institute for Cancer Research, Wellcome Sanger Institute, Broad Institute of MIT, and Harvard and MIT. And EMBL EBI, and we have an international core funders forum with funding to support our activities on Secretariat from NIH, Wellcome, Genome Canada, CIHR. And then next, this is our leadership team. We have 4 members of the GA4GH Executive Committee and somewhere around 48 or so individuals on the GA4GH steering committee, which approves all the standards. I am not going to play this video now. Stephanie Lee, our creative lead extraordinaire, is going to pop some links into the chat to watch this video in English, or if you are so inclined, in Japanese. We are going to be translating it into a number of additional languages soon, so stand by for those. I hope if I click forward, it won't start the video. Justina, let's see. Oh, shoot, okay. All right, okay. Jeremy and maybe Lindsay, I might ask you guys to turn your videos on so we can answer questions together, and anybody else that wants to, Nirja too, sure, please. So let's just go through them. You can also upvote them if you're interested in somebody else's question. Does GA4GH work with other global human rights organizations like the UN, FIDH, et cetera? So no, not as far as I'm aware with human rights organizations. We do work with the WHO a good bit and the World Economic Forum, but specifically the human rights activities are really the brainchild of one of our founding RUSE leads, Bartha Knoppers at the Center for Genomics Policy. But that's a great idea and probably something we should follow up on. Any insurance companies involved as partners? Very good question. I do not know of any. Lindsay or Jeremy or Nirja? No.
No, not that I'm aware of.
Okay. But also another really good point that these are definitely communities that, you know, GA4GH was born in the research sector. And as I was saying, the genomics data coming more and more from the healthcare and clinical community, like we really do need to start partnering with those sorts of organizations more. And so if you are coming from that sector, we would love to have you on board. A learning health system relies on good EHR integration. Genomics and EHRs aren't very mature. Getting cloud providers on board is good, but you also need Epic. This is a comment. Does anybody want to respond to the comment?
I can just maybe add in quickly. Yes, you know, it'd be great to have, you know, better EHR vendor participation, and you know, we would certainly welcome everyone to get involved. I think our main engagement with a lot of EHR vendors is actually going to be via HL7 and FHIR. We do have a couple of efforts to translate some of our standards into FHIR implementation guides. And so some of them are almost ready to be implemented. But yeah, I think that's going to be our main path for better integration. I don't know if anyone else wanted to add anything to that.
No, you're the resident expert there, I think, Lindsay. So this next one was answered in the chat. For phenotypes, do you focus on human relevant phenotypes only or other models species too? As Lindsay said in the chat, it's mostly humans. That's the driving use case, but it might be applicable, you know, if you're interested in using it elsewhere. And then finally, do the gene expression standards include transcriptional isoforms? If so, which reference models does GA4GH recommend? RefSeq, Ensemble, AceView. Okay, admitting complete ignorance here. I know that I hear about Ensemble a lot more than the other two, but Jeremy or Lindsay, do you guys have a better answer here?
I can comment a little bit, but I don't have a definitive answer. So this kind of falls into the RNA get standard, as well as there's a sequence annotation standard within the genomic knowledge standards workstream. And so they are developing a model of sequence annotation. So annotating it with, you know, transcription, start and end sites and isoforms and splice forms and things like that. And it should be leveraging the existing standards and models that are already in this area, like RefSeq and Ensemble. So I don't work with that work stream directly, but that would be where that work is happening. So if this individual is interested in seeing how a GA4GH standard can come out of those existing tools, I'd recommend joining the sequence annotation working group.
Right, which is a part, I don't know if you said this, a part of the genomic knowledge standards work stream. So I think Amreen might be putting a link in the chat for an exit survey here, but I think it's also worth just to let us know how you feel this event worked for you, what you got, what you didn't get, what would you like to see if you were, you know, joining again in the future. But I think we can also, you know, we want your feedback about this event, but we also want you to reach out if you want more information about any of these questions. So Jeremy, Lindsay, Nirja, and I are only 4 of a 20+ person staff team. And as you can tell, Lindsay's very focused on the clinical and phenotypic data capture work side. I mean, that's what her work stream she manages along with the RUSE, and Jeremy's on the technical team, and Nirja and I are kind of, you know, all across all of it. So if there is a specific question, you know, related to something more, you know, a deeper specificity that you're looking for, reach out, secretariat@ga4gh.org or info@ga4gh.org, either of those works. And we will find somebody to answer your burning questions. I think that's our last slide. Yes. So yeah, we've got 2 minutes left. If there are any other questions or if anybody wants to raise their hand and chime in, feel free. Otherwise, we will give you back 2 beautiful minutes. Thank you, Andrew. Thank you for all of your great questions. And do reach out if you want to learn more. All right. Well, that's a wrap. Have a great evening or day if you're calling from or joining from Australia, and we will hope to see you on the work streams.